Okay, the second half of our video. Now this is going to be detailed, and then I'll have to show you a slide on your PowerPoint that's really busy, but it's got all the answers for the last part of this. These are called, this is the steps of a muscle contraction. Now, this is written verbatim for you, so there's nothing I'm going to leave off, nothing that you're going to miss or anything. Now, I do want to say one thing. When a muscle fiber is relaxed, you can see the troponin is blocking the binding sites on actin, and it's preventing that little myosin head from attaching. That's a resting muscle. That's why it's at rest. It's not contracting. So just to a real quick review, remember, if you get down on the cellular level, this is a thick filament. It's got all these little myosin heads, and there's a point during a muscle contraction where the little head will reach up, it will grab, and it will pull on a thin filament. Now, on your, I apologize, I'm having a meathead moment. Hold on, let me go back. There we go. Let me go back to our class. Pregnant pause. What a summer. Let's see. Yeah, under the overview. Now these videos will be above that. I apologize for that. I was going to say, I gave you these diagrams. Now these are pictures you can click on. So here's one, and then the other one. Okay, it kind of goes through all of it together as well. I'm going to be using this one that says muscle contraction steps. This is a great picture. It breaks it down to 6 really easy steps. But I thought I had given you, but that's fine. I thought I had given you another one. But this one does kind of show, look, see this myosin head? It's grabbing and it's pulling. See the arrows? So when a muscle's relaxed, that can't happen. So hence a relaxed muscle. But so a relaxed muscle, here's the tropomyosin, and it's blocking that little binding site on actin for that little head. And they have a strong affinity for each other. So that's kind of where you start. The whole thing sort of teeters on that. All right. So let's go through all the steps. And I'm going to go back to, at some point, I'm going to go back to this. I'm going to go ahead and let you look at those two. Those are really good. Oh, there we go. I don't know what's going on there. So we got bigger, smaller. There we go. I'm going to go back to this. I'm going to stay on this document. You can look at the others. They're really good. They're a little more detailed. So when a nerve impulse reaches the neuromuscular junction, we covered that in the last video, a chemical neurotransmitter called acetylcholine is released. This chemical carries the stimulus across the gap between the neuron and the muscle cells. So that was actually called the synaptic cleft. So here is the terminal axon, and you can see there's the ACH being released, and remember it's going to come down here and bind to little receptors in the neuromuscular junction. Here's the sarcolemma, and when it does that, it's going to depolarize that area, and that wave of depolarization is going to start that way. You're going to have sodium ions rushing into the cell, the charge temporarily reversing across the membrane, and then right behind it's going to repolarize. So it says right here, neurotransmitter releases diffuses across the synaptic cleft and attaches to ACH receptors on the sarcolemma. And that part that's folded, it's actually called the motor end plate, but it's the folded sarcolemma. Acetylcholine will result in depolarization of the muscle fiber membrane. And the excitation of the membrane is transmitted via the T-tubule system into the sarcoplasmic reticulum inside the cell. So this is supposed to be one of those transverse tubules. And remember, the cisternae in the last video of the sarcoplasmic reticulum, it stores calcium ions. So if we're going to go back to our picture, that signal's traveling this way, and it comes down the first transverse or T-tubule, And you can see when it does that, that is the stimulus to release calcium ions, the red dots. See, it's flooding out, and these are called the cisternae of the sarcoplasmic reticulum. I covered that in the last video. Okay, so the above activity results in the transient release of calcium ions from the sarcoplasmic reticulum. So that's what that's showing as well. See the calcium being released. Calcium ions activate the actin myosin to bind in the following manner. Now, remember, I had covered, there's a part called troponin that binds to calcium ions on a thin filament. When a muscle cell is relaxed, the cross bridges of myosin, the myosin heads, are unable to bind to actin. Okay, I just did that a while ago. So, like I said, right here, this would be a relaxed muscle. See how the tropomyosin is blocking that little binding site? Oh, and while I have it here, so here's troponin, okay? And there's actually 3 troponins. I'll leave that for another day. There's a troponin I, troponin T, and troponin C. And troponin C is the one that binds to calcium. And if you're in the medical field, troponin is an indicator for a heart attack because cardiac cells start to burst, and you look for troponin I as a blood marker for a heart attack as well. Okay, so the inhibition of cross-bridge binding is due to 2 proteins, troponin and tropomyosin. When calcium binds to a specific site on troponin, troponin C, it changes the shape of the molecule, causing it to pull tropomyosin to one side, uncovering the binding site or active site. So see here, it's covered up. Once calcium binds there, do you see how now the tropomyosin's been moved over? It says active sites exposed, and the little heads reach up, they grab, and they pull. So, uncovering the attachment site with ATP for energy, myosin pulls actin toward the center of the sarcomere. Remember, when muscles contract, they develop tension, they shorten. And this is it's called the sliding filament theory. These little filaments are sliding past each other. As the sarcomeres shorten, the entire muscle fiber shortens. And that's because of that pulling right there. Okay, so you have an action potential. The action potential travels down the transverse tubule. It causes calcium to be released. Calcium goes down. It binds to troponin. Troponin pulls over tropomyosin, exposes the binding site. That little head immediately reaches up, grabs, and it pulls. All one motion. If you go back to our very simple diagram that I would use to study for your exam, so we're right here at number 3, calcium ions bind to troponin. Troponin changes the shape, removing the blocking action of tropomyosin, and the active sites exposed. See the little thing grabbing and pulling. It has contraction. Myosin cross bridges alternatively attach and detach. And there's a video you can look at. When you look at this video, you'll see one, and I'll have it labeled. It has to be a 7th grader. They made a video called 12 Steps of a Muscle Contraction. It shows this very well. It's a very simple video. It'll be a great thing for you to look at when you're studying. Pulling the actin filaments toward the center of the sarcomere, release of energy of ATP powers this whole process. So here's a point where if you were in class with me, I'd ask you, can that little head stay attached here? Can a person walk around in a permanent state of contraction? Only if you're a high school teenager who plays football. They kind of walk around in a permanent state of partial contraction. But I'm kidding, but no, of course you can't. A muscle cannot stay contracted. At some point, it's got to relax. So what happens is this calcium ions, once it binds here and causes that tropomyosin to be moved over and the little thing grabs and pulls, well, calcium is pumped back where it came from. Like it says here, removal of calcium by active transport into the SR after the action potential ends. And what happens next is ATP stored in muscle actually binds to that little myosin head, causing it to detach, and then it moves the head back up it sort of recocks the position. And that's why it's showing it uses ATP. It splits an ATP molecule for energy to do that. And that's step number 6. And then the whole thing would start all over again. So when a muscle contracts, steps 1 through 6, that's what happens. And it's a cyclic activity. As long as there's a stimulus, it would keep doing that. So down here, the aftermath, a calcium pump returns calcium ions to the sarcoplasmic reticulum, which allows the troponin tropomyosin protein to cover the binding site on actin. So the myosin head can't attach anymore. Filaments return to their positions, and the sarcomeres return to their original length, and the muscle cell relaxes. Now, if you're doing constant lifting, that whole thing is going on the entire time. But at some point, yes, you're going to relax a muscle. These right here are talking about how does the muscle fiber relax. Acetylcholinesterase. Now, I know there's a blank here, but that's actually an enzyme that breaks down acetylcholine. And that picture is actually in your PowerPoint, but let me, this information is going to be in your PowerPoint. And I apologize for the small font. But see this? Look, here it's attaching, pulling, detaching, recocking, attaching, pulling, as long as the stimulus is there. When it goes to relax, so here's acetylcholinesterase. Acetylcholinesterase is an enzyme found in the neuromuscular junction. I showed you that earlier. It breaks down acetylcholine, so it removes the stimulus. So that's one way. Calcium is pumped back to the cisternae of the sarcoplasmic reticulum. I just showed you that. So after it binds to troponin, calcium is pumped back to where it comes from, and then ATP binds to the myosin heads. Like I just mentioned that earlier. It actually splits the ATP to recock the myosin heads just in case there's another contraction. But those 3 mechanisms allow a muscle to relax. And it kind of says that here, calcium is pumped back, ATP binds to myosin heads. What are some sources of energy for a muscle to contract? Once again, I apologize for the busy slide. See, this just breaks all kinds of rules, but here are 4 sources of energy for a muscle contraction. There's already some ATP stored in muscle, not to gross you out, but just next time you buy a hamburger or steak, you notice how it's got a red color to it. That's because it's packed full of oxygen. There's ATP, there's myoglobin in muscle, and there's ATP stored in muscle. Muscle has the ability to do that. So that lasts for a very few seconds. That just gets the entire process started. There's a pathway called creatine phosphate. You can buy creatine in GNC. Creatine phosphate is a high energy substance. It's used for high intensity exercise, weightlifting, sprinting. It lasts about 10 seconds, but it provides energy for those little heads to grab and pull during high intensity exercise. Okay, so you're not going to be using the creatine phosphate pathway while you're walking or jogging. All right, that's for high intensity exercise. The last 2 you've probably heard of. Anaerobic respiration is a pathway. That's what you call those in biochemistry. It is also for moderate to high intensity exercise. It's basically where the body is using a different energy source without oxygen as part of the process. It doesn't mean you're not breathing, but it does not use oxygen to produce ATP. And once again, it has toxic byproducts, such as lactic acid. It says that there. But this is for moderate to high intensity exercise. Just think of, I ran track in my youth, but people who run a quarter mile or the 800 meter dash or something that is a little higher intensity than just walking or jogging. That's the anaerobic pathway. Aerobic respiration is using oxygen to convert energy, and it's a lot of chemical reactions. It's what's used during low intensity exercise or daily living activities. It has water and carbon dioxide as byproducts. Like it says here, carbon dioxide and water molecules are byproducts of that. But if you want to know a good way of determining what pathway you're in, if you're walking and you're walking with someone and you're carrying on a conversation, or maybe you're jogging and you're carrying on a conversation, if you can talk while you're doing your exercise, you're in what's called the aerobic pathway. If you started running faster or walking faster or getting fatigued, and you're getting to the point where you cannot carry on a conversation, you're now switching to the anaerobic pathway. Your body's using a different energy source to produce ATP. And yeah, anaerobic will eventually slow down. Lactic acid will fatigue a muscle. And, it depends on your fitness level as well. All right, last thing here, and this is a really detailed section, but it says here there are over 600 muscles. I want to say there's 734. Depends on what book you read, but a little bit about muscle terminology. Now, that's a good picture. I think I have a better one here. Well, I do, kind of. I thought I had a guy lifting a weight. I guess I dreamed that one up. Well, anyway. Okay, so all muscles have an origin and what is called an insertion. This is the radiotuberosity. This is the bicep. This is called the coracoid process of the scapula. The origin does not move. The insertion is the part when a muscle contracts, it pulls the insertion closer to the origin. And all muscles have that. The body is the part of the muscle that's attached between the two of those. But any kind of action you do, the prime mover, if you're flexing your bicep, the bicep is called the agonist or the prime mover. The triceps in the back of the arm would be the antagonist. It relaxes and allows the prime mover to contract. Your brain has something called reciprocal innervation. Your muscles do. But your brain relaxes part of your muscles and contracts the others. That's what tetanus blocks. Tetanus will block what's called the reciprocation pathways to where all the muscles contract, and then a person can't move, they can't breathe, they can't do anything. So the antagonist is the muscle that relaxes as the prime mover is contracting. And a synergist is any muscle that helps stabilize the prime mover. Like that person's doing a bicep curl. There's a muscle called the brachioradialis and the brachialis. Those are also allowing the bicep to contract. So those are all terms of muscles. And yes, it's a detailed section. Please check out all the videos I have posted for this under this particular tab. And I hope this helps. And thanks for joining me.
And that's the bottom line, because Stone Cold